Morning, everyone. Colleagues at the head table. Before I get into the presentation, I think by now what should have been said was that I had very little to do with the actual work. It's my colleagues, Doctors Kafra, Buddy, and Murphy from the UK. But the way it works in CABI is if you're in a project, you have to be paid. And I guess we didn't get enough money from the EU for me to be paid to do anything on the project. So I'm working for free today by just presenting this. But before I get into the presentation, I just want to share a bit of experience and comment a little bit of what was said this morning. Recently, in another project that CABI is working on called PlantWise, where we look at pest and disease problems of farmers in the field and try and solve that, and again with the aim of improving food security, reducing losses, etc. We looked at Christophine in the Northern Range, and something very unique is with Christophine production in Trinidad is it tends to be seasonal, and the lowest yields are in the wet season. When you look at significant producers of Christophine, Costa Rica, Mexico, Guatemala, Jamaica to some extent, Brazil and so on, it is never seasonal in those places. So one of the possibilities is that we have a pollination issue with Christophine as well. So it's just to make the point of what was said this morning, that it is probably a bigger problem, and what was learned here can certainly be transferred to other crops in the region. So to get to the presentation, basically, I'm presenting on behalf of my colleagues, and this presentation focus a little bit on CABI's microbial and molecular services. I'll give an overview, some typical areas of work and specialisms, DNA barcoding, what it is, insect barcoding, and what we actually did with the cocoa project. So CABI has a staff of about 19 based in the UK that provide a wide variety of services and biological resources to commercial customers, CABI projects, and CABI member country organizations. CABI as an organization is basically a member country organization with 48 member countries globally. And what the CABI microbial and molecular services do is they do DNA sequence-based identification on microorganisms. They provide environmental and industrial services, supply, preserve, and safe storage of microorganisms. They do contract research, training courses, for example, in advanced plant pathology techniques and so on, and of course, publications on special issues relating to microbial and molecular work. Typically, typical work areas of this unit would serve a wide array of industries. So for example, in agriculture, they provide crop pathogen identifications. For the food and industry, they provide product contaminant identifications. For the transport industry, microbial contaminant assays in fuels, and we have Conidia, which is a spin-off company from CABI that provides, build, and supply test kits for aviation fuel, which has turned out to be quite something significant. In terms of the environment, we do air and building surveys for fungal contamination. In terms of the electronics industry, resistance to mold growth testing. In biotechnology, patent deposits and safe deposits. Drug discovery, culture and extract provision. For the pharmaceutical industry, production, environmental monitoring. And in terms of academia, identifications, culture supply and training. And the biological resource cultures, they supply culture and even train in terms of identifying, growing, and everything else to do with them. And then specialist mycology publications. In terms of microbial identifications, we have the bacterial identification by partial 16S ribosomal DNA sequencing, which has been accredited since 2012 by the ISO 17025 and ISO 90001 by the UK Accreditation Service. fungal and yeast identification by internal transcribe spacer, and other generic specific loci sequencing. And this has been accredited since 2013 by ISO 17025 by, again, the UK-based accreditation service. With respect to the environmental and industrial, services are accredited by the UKS include resistant to mold growth testing of electronic equipment and components in accordance with that standard there, assessment of microbial contamination of fuels and lubricants in according with the IP 385-99 Procedure A standard. And when CABI refers to fungi, what was traditionally studied by mycologists as fungi, including the filamentous organisms and oomycetes, things like Pythium and Phytophthora and so on. In terms of the microbial services, or resources rather, CABI hosts a collection of approximately 28 living cultures, 28,000 living cultures. These are primarily fungi, but some bacteria are also included. We also host a number of smaller non-CABI collections, and these are, for example, the Antarctic Survey Collection of fungi and bacteria, and the UK National Collection of wood-rotting fungi. The preserve formats include oil immersion, freeze drying, liquid nitrogen, cryopreservation. We supply cultures and extracts, including validated reference cultures, and their patent and safe deposit services are also available. Compliant with the spirit of the CBD and the Nagoya Protocol. I'm sure you all are aware of the CBD and in terms of access and benefit sharing. And basically, this 28,000 living cultures are basically collected in member country states from 48 countries around the globe. So therefore, CABI actually basically maintain these on behalf of the countries. So something may not be from a country, but nobody can find a pure strain of it anymore. It is kept in the UK, where it is more economical to do so. With respect to insect barcoding, or did I go through that properly? Yeah. With respect to insect barcoding, this is something relatively new for CABI. In 2015, we implemented, again, the UKS accredited protocols, and we applied standard methods to barcode insects. And the development of DNA isolation using PCR sequencing of COX1 region and of the mitochondrial DNA. Insect specimens collected in ethanol produced a positive PCR product, and collection into ethanol was the optimal collection method. So this obtained very good quality amplicon and DNA sequences. And if you need to know, the COX1 is the internationally recognized DNA barcode for all values of the animal kingdom except nematodes and a few taxonomic groups. How to barcode insects. Basically, four main aspects could be used, or four main sources. We obtain them from our specimens, from museum collections, and from nature. Laboratory analysis using standardized protocols. And the third one, which is you must have a database of DNA data. And this is possibly CABI's strength, because we do have this tremendous collection of over 28,000 of what has been identified, so that you can then do the DNA sequencing of those and then compare it to what you have in nature, because it's quite easy to do a DNA sequence of any random species in nature. But if you don't have a standard to compare it to, that is where the trick lies. And that is CABI's strength, because we have this huge collection that now we've started actually doing the DNA sequencing for. It means that we're going to have a huge strength in this area. So then data analysis of the DNA barcode is then matched with this known database, and that makes life a lot easy. Especially, so why do we need to barcode